In our previous video regarding transcription in eukaryotes, we have seen that the initiation of transcription, it involves many transcription factors, and similarly even the termination of transcription in eukaryotes, it is a complex process which involves various proteins and enzymes. Now, basically, as we know that in eukaryotes, there are three types of RNA polymerases that are involved: the RNA polymerase first, second, and third. And they are used for the transcription of the three different types of RNAs, that is the ribosomal, the messenger, and the transfer RNAs. The termination process in all the three cases is different. For the purpose of polymerase first and RNA polymerase third, it is basically a very simple process. It may involve some transcription termination factors which may cause the cleavage at the end of the transcript. However, in case of RNA polymerase second, which is responsible for the transcription of messenger RNAs, the process of termination is a bit more complex. Now, there's a reason for it. The reason is that the product of RNA polymerase second is the RNA that is basically the messenger RNA. And in case of eukaryotes, this messenger RNA, which is produced within the nucleus, and it transcripts, or it is the transcribed part of both the exons and introns. It has to undergo processing and it has to be maintained until and unless it reaches the ribosomal machinery present in the cytoplasm. So, along with the transcription, there has to be the protection of the 5' end. That is, the 5' capping has to be there. And at the time of termination, a poly A tail has to be added at the 3' end of this transcript. The other part, that is splicing, it is another process. But basically at the time of transcription, the capping and tailing, they have to take place. So the termination part of transcription of this messenger RNA is a bit more complex. This termination, it basically involves first the cleavage at the end of this gene sequence, where this poly A tail has to be added, and after that removal of this transcription bubble, which is made up of the DNA molecule along with this transcript bubble. This is the elongation complex, and here they are this transcript bubble, it comprises of say 30 to 60 nucleotides where the complementary base pairing is not there, and in this region the new ribonucleotides, they are polymerized, resulting in the formation of this RNA. And approximately a length of 8 to 10 nucleotides of RNA, they form a hybrid with the DNA template. So at the time of termination, this hybrid part, it has to be cleaved, it has to be disrupted, and then this RNA polymerase, it has to be removed from this DNA double helix duplex molecule. So ultimately, this transcription termination in eukaryotes is a bit more complex a process. Now, as we know that at the time of initiation of transcription in case of eukaryotes, a pre-initiation complex is formed by the various transcription factors at the promoter, and then after phosphorylation of the C-terminal domain of this RNA polymerase, it takes place by this transcription factor second H, kinase activity of this transcription factor. This phosphorylation is basically at the 5-serine of the CTD. And once this phosphorylation takes place, it leaves the promoter, it escapes this RNA polymerase second, it escapes the promoter region. And as the elongation progresses, there is a phosphorylation of CTD and dephosphorylation of some other elements of this CTD region. But prior to that, at about 20 to 40 nucleotide sequences downstream from the promoter, this RNA polymerase, it stops the elongation process. That is because there are two regulators of this transcription process, that is DSEF, and the negative regulator of this transcription, that is the NELF. These two are also associated with this RNA polymerase second. And in order to initiate or to carry on the process of transcription beyond this point where it aborts, phosphorylation of these two regulator proteins is essential. And this phosphorylation of DSIF and the NELF, which is the negative elongation factor, that is carried over by a protein that is PTEFB. This PTEFB, it phosphorylates both the DSIF and the NELF. Now, when this DSIF gets phosphorylated, it induces elongation, whereas this negative elongation factor, that is NELF, it disassociates from this complex, and thus it promotes the transcription or elongation of this transcript. Along with this factor is also responsible for the, it has got the kinase activity, and it is responsible for phosphorylation of different residues of the C-terminal domain of RNA polymerase second. Now, there's a sequence of phosphorylation and dephosphorylation of various residues. Initially, at the initiation of this transcription, the serine 5, it is phosphorylated, and gradually as the elongation progresses, it dephosphorylates with the help of enzyme phosphatase. Simultaneously, the serine 2p, this serine 2, it gets phosphorylated. So the amount of serine 5 phosphorylation, it decreases as one moves ahead on this elongation process, whereas the amount of serine 2 it increases, and there's a peak when it reaches the termination region of the phosphorylation of serine 2 residues. And then after, once it moves past this termination point, there's a marked uh, drop in the phosphorylation of this serine 2. Beyond this, um, this poly A signal, that is the from where this poly A tail it has to be added on the transcript. Apart from that, the tyrosine 1, it also increases its phosphorylation, it also increases, but just before this poly A signal, it abruptly it stops, it drops, and phosphorylation of this tyrosine 4 it occurs just after this poly A um, signal. So in this way, phosphorylation of serine and tyrosine, different residues, that it is a dynamic process that takes place during the elongation, and as it nears this poly A signal, it the phosphorylation it signals that now there's the poly A signal or the pass has arrived, and now it has to the transcript it has to be uh, truncated. So after this, how it truncates? The pass or the poly A signal, just prior to it there is a cleavage site. This cleavage site is conserved. It consists of AAU-AA sequence and approximately 40 to 60 nucleotides downstream. There is a poly-U or GU rich sequence, and the exact site of cleavage it lies somewhere between these two consensus sequences where the cleavage will take place, and it is approximately 10 to 30 nucleotides downstream this AAUAA sequence. Now on the tail of the polymerase enzyme, certain other factors they also are present, and two such factors, they are the CPSF or the cleavage and polyadenylation specificity factor, and the CSTF, that is cleavage stimulation factor. They are basically present on the tail of poly A polymerase, RNA polymerase. So on this, these two factors they are transferred to the new messenger RNA transcript. Once its sequence, the poly A signal from where the cleavage site it has to be cleaved, that part of DNA, once it is transcribed, these two factors they are transferred from the CTD to the nascent RNA formed, and they attach to these points. The CPSF it attaches to AAUAA, whereas this U or GU rich sequence it provides the site of attachment for this cleavage stimulation factor, and it brings about the cut of cleavage of this pre-messenger RNA, and at this cleavage, the poly A tail has to be added. So now two more things they remain. First, addition of the poly A tail, and then after this RNA polymerase, it has to be set free from this DNA. Then only the termination will be completed. So basically this CPSF, which is also known as the CSPF, this factor, it has got two domains, the 160 domain and the 73 domain. Domain, we know it is the site or the part of the protein of the enzyme. So this 160 domain, it is used for the attachment of this SPSF to this AAUAAA sequence, whereas this 73 domain, it is responsible for the endonuclease activity, and it breaks the phosphodiester bond of the messenger RNA that is being transcribed at the cleavage site. It cleaves it, and once it cleaves it, this poly A polymerase enzyme that is responsible for the formation of poly A tail, it gets activated, it starts its activity at the point of this cut region, at the 3' end of the newly formed messenger RNA. Now, the characteristic feature of this poly A polymerase is that it do not require any template. Only it uses the adenosine triphosphate molecules and adds adenine one at a time, and this tail, it may range up to about 300 adenine nucleotide, and as this tail is being formed, it has to be protected. It has to be protected so that it does not form the double stranded structure with the existing poly U sequences on this messenger RNA that has been transcribed. So it is protected by single strand binding proteins, the poly A binding proteins, the nuclear version of these PABs. And when this final messenger RNA, it will reach the cytoplasm after being processed. At that point of time, this nuclear poly A binding proteins, they will be replaced by the cytoplasmic version. So N/C is written over here. Now this how the poly A tail is added upon the newly formed messenger RNA transcript. Now one more process remains, and that is a removal of this RNA polymerase. And as we are seeing that the cut is in between these two factors, that is the CPSF and the CSTF, in between these two factors. So removal of this RNA polymerase, it may take place by any of the two mechanisms, or rather we may say that there are two hypotheses to explain the removal of this enzyme from the DNA double helix, from opening up of this transcript bubble. This disengaging of RNA polymerase, it may be either by means of allosteric termination, that is, some termination factors they may be involved, And once these termination factors, they get associated with the RNA polymerase second after it passes the poly A site or the cleavage site, they induces some conformational change in the structure of the RNA polymerase, and that destabilizes this enzyme and it separates or disengages from the DNA double helix. Second hypothesis says that it involves exonuclease activity, and that model is known as the torpedo model. According to this model, once the cleavage, it occurs at the cleavage site. Now the 5' end, of the messenger RNA that is being formed, it has already been capped during the process of elongation. But once the cleavage takes place at the cleavage site, this 5' end is uh, exposed. It is exposed to various exonucleases. And in case of human beings, the exonuclease involved is this XRN2. So this exonuclease, it starts degrading the remaining part of the RNA, because RNA polymerase, it does not stop its activity at the cleavage site. It keeps on transcribing down the DNA molecule, and the RNA that is being formed, it is simultaneously being degraded by this exonuclease. And as this exonuclease, it reaches and comes in contact with this RNA polymerase, it induces some conformational changes. And because of that, as it uh, degrades the RNA, this hybrid, it dissolves because now no RNA is there, and conformational change take place in this RNA polymerase, resulting in its disengagement or its removal from the double helix, and normal double helical structure is regained. So in this manner, the RNA polymerase second, it not only processes the or adds the poly A tail to the nascent messenger RNA formed, but it also involves disengagement of the RNA polymerase from the DNA double helical structure. And because of this, the termination in eukaryotes of the messenger RNA, of the pre-messenger RNA that is formed, by the time it finishes, capping is already done, that is addition of the guanyl cap at the 5' end. The tailing, the poly A tail is there at the time of termination, and the remaining part of processing, that is splicing, that is another topic that we will deal in the other video. Thank you.